Enzymes consist of three main structures: primary, secondary, and tertiary structures. The primary structure is a chain of amino acids bonded together by peptide bonds. This strand of amino acids create the protein that the enzyme is made up of. The secondary structure has two different kinds of structures. One is a helix and the other is a sheet. This happens because the hydrogen in the enzyme, NH3, and the carboxyl group, COOH, bond together, causing the strand of amino acids to fold over each other. Finally, the structure, the tertiary structure, is the actual folding of the proteins, giving the enzyme half a 3D group. All enzymes are catalysts in chemical reactions, which means that they speed up reactions without changing any of the chemical properties of the reaction. The most common place to find bromelain is in the pineapple fruit, more specifically the stem of it. Bromelain is a natural anticoagulant that breaks down fibrin, the protein that clots blood. This in return thins blood and thins mucus. Also, bromelain triggers change in white blood cells that helps improve the overall immune function of the organism it is exposed to. The enzyme also has strong anti-inflammatory properties that help healing of small muscle injuries, i.e. strains and sprains.
The active site of an enzyme is where the substrate bonds with the enzyme. The active site is created by a few amino acids that create a groove in the enzyme surface. The substrate is the substance that the enzyme bonds with to make the substance more reactive. The active site and the substrate work together like a lock and key. Only the enzyme that is supposed to bond with the substrate has the same shaped groove that the substrate can fit into perfectly. Environmental factors such as temperature, chemical agents, and pH changes cause denaturation of an enzyme. Denaturation is when the enzyme loses its 3D shape of a protein. By losing its shape, this causes the active site to be destroyed, which makes the enzyme inactive. Competitive inhibitors prevent the formation of enzyme substrate complexes. They do this because they are similar shape to the substrate, which means they can fit into the active site. The inhibitors remain unbonded because they have a different structure than the substrate, allowing less substrates to bond to the enzymes. Non-competitive inhibitors prevent enzyme product complexes to form. These inhibitors bind to the enzyme allocentric site. This destroys the tertiary structure, which in return does not allow the enzyme to work as a catalyst. As many inhibitors can have a very negative effect on the organism they're exposed to, they also have their benefits. Some inhibitors are used in medicine. As most of these inhibitors are competitive inhibitors, viruses cannot build protein coats, which in return makes them unable to replicate.